today we are going to talk about the animal development and aging. So basically, where the human comes from. We comes from the fusion between the sperm and an ovum to form a zygote. Before I proceed into explaining the fertilization process, let me explain about the structure of the sperm and the structure of an ovum. Okay, so sperm can be divided into three parts, which is the head, the tail, and the middle part. The head of the sperm contains haploid nucleus and is kept by a membrane-bound acrosome that secretes enzyme. The middle part of the sperm contains mitochondria that produce ATP. By producing ATP, it will generate energy to help the sperm to swim through the fallopian tube. And for the tail, sperm contains a flagellum tail that allows it to swim. So that is for the sperm structure. And now let me explain about the egg. The egg of a mammalian is surrounded by a few layers of adhering follicular cells called the corona radiata. These cells nourish the egg when it is in a follicle of the ovary. The egg of the mammalian also has extracellular matrix that is called zona pellucida, which located outside the plasma membrane, just beneath the corona radiata. So that is for the structure of the ovum and the sperm. Now let me explain about the fertilization process. Fertilization process is basically where the fusion of the sperm and the oocyte occurs. Fertilization requires several steps that will result in the diploid zygote. Fertilization started with millions of sperms that swim through the fallopian tube, and only hundreds of them will succeed in reaching the oocyte. Isn't it amazing, the fact that we are one of the fastest swimmers way back then? So let's continue with how the sperm can enter the oocyte. So during the fertilization, the sperm cover the surface of the oocyte and release enzyme to weaken the corona radiata. They squeeze through the corona radiata and bind to the zona pellucida. After a sperm head binds tightly to the zona pellucida, the acrosome releases enzymes that forge a pathway for the sperm through the zona pellucida to the oocyte plasma membrane. After a sperm has bind to the plasma membrane, polyspermy occur, which block the entrance of other sperm. As soon as sperm touch an oocyte's plasma membrane, the plasma membrane depolarizes, and this change in charge, which is called the fast block. It repels sperm for a few seconds. Then, vesicles in the egg, called the cortical granules, secrete enzymes that turn the zona pellucida into impenetrable fertilization membrane. The last event of fertilization include formation of a diploid zygote. Microvilli extending from the oocyte's plasma membrane bring the entire sperm into the oocyte. Sperm nucleus releases its chromatin, which reform into chromosome. The secondary oocytes become an egg. A single nuclear envelope soon surround. So that is all my explanation about the fertilization process. So now let my friend explain about the embryonic development.
So hello, and let's move on to the embryonic development. So you've heard about what happened during fertilization in animals. So we know that when a sperm fuses with an ovum, it will produce a zygote. So shortly after fertilization, this zygote will undergo a cleavage and resulting in multicellular embryo. DNA replication and cell division will occur repeatedly, and cells will eventually get smaller with each division. And from a zygote, it will continue to divide and divide and divide and divide and form a blastula, a hollow ball of cells having a fluid cavity called a blastocele. So let's take a chicken for example. For a chicken, it lays a hard-shelled egg containing yolks. So please note that yolk does not involve in the cleavage. So what happens is that when blastocele forms, it separates the yolk and the blastocells. So for a vertebrate zygote, it also forms a morula, but it differs from a chicken. When there's yolk, the zygote and the embryo exhibit polarity, and the embryo has an animal pole and a vegetal pole. So the animal pole will develop into the ectoderm and the endoderm, while the vegetal pole will develop into the endoderm. So that's the end of cellular stages.
So now we move on to the next stage in this embryonic development, which is the tissue stage. So the tissue stage involves gastrulation, which is the formation of gastrula from the blastula. So there are two stages in gastrulation, which is the early gastrula and the late gastrula. The early gastrula stage begins when certain cells begin to invaginate the blastocele to form two layers of cells, which is the ectoderm and the endoderm. The invagination of cells into blastocele will form a blastopore, and in a lancelet, the blastopore will eventually become the anus.
So late gastrulation occurred differently in a lancelet, a frog, and a chick. In a lancelet, mesoderm formation begins as outpocketings from the preventive gut. These outpocketings grow in size until they meet and fuse, forming two layers of mesoderm. The space between them is the coelom. The coelom is a body cavity lined by mesoderm that contains internal organs. So in humans, the coelom becomes the thoracic and abdominal cavities of the body. So in the frog, cells containing yolk do not participate in the gastrulation, and therefore they do not invaginate. Instead, a slit-like blastopore is formed when the animal pole cell begins to invaginate from above, forming endoderm. Animal pole cells also move down over the yolk to invaginate from below. Some yolk cells, which remain temporarily in the region of the blastopore, are called the yolk plug. Mesoderm forms when cells migrate between the ectoderm and endoderm. Later, the mesoderm splits, creating the coelom. In a chicken egg contains so much yolk that endoderm formation does not occur by invagination. Instead, an upper layer of cells becomes ectoderm and a lower layer becomes endoderm. Mesoderm rises by an invagination of cells along the edges of a longitudinal furrow in the midline of the embryo. Because of its appearance, this furrow is called the primitive strip. Later, the newly formed mesoderm splits to produce a coelomic cavity.
So the ectoderm, endoderm, and mesoderm is called the three embryonic germ layers. So it doesn't matter how gastrulation takes place, it will always give you the same result, where three layers will be produced.
So now we have finally reached the last stage in embryonic development, which is the cellular stage. So in cellular stage, the newly formed mesoderm cells lie along the main longitudinal axis of the animal will merge to form a dorsal notochord. The notochord will continue to develop in lancelet, but in a frog, chicks, and human, it is replaced with a vertebral column, which is why we are called the vertebrates. The nervous system develops from midline ectoderm located just above the notochord. At first, the cells on the dorsal surface of embryo thicken, forming neural plate. Then, neural folds develop on either side of a neural groove, which becomes the neural tube when the folds fuse. At this point, it is now called a neurula. Later, the interior end of the neural tube develops into the brain, and the rest will become the spinal cord. Midline mesoderm cells that did not contribute to the formation of the notochord will now become two longitudinal masses of tissue. The two tissue masses become blocked off into somites, which are serially arranged along both sides along the length of the notochord. Lateral to the somites, the mesoderm splits, forming the mesodermal lining of the coelom. So from that, organ formation continues until germ layers have given rise to the specific organs. So we have reached the end of embryonic development. So that's how an embryo is developed.
So I guess we have reached the end of our video. I hope you guys enjoyed this video and you can get a better understanding for this topic.
And don't forget to give us a thumbs up and leave a good comment below. Bye bye!